Good afternoon, everybody. It's my true pleasure to welcome you to the Associated Professor Lecture within the strategic program Cancer. My name is Jürgen Kubal. I'm a hematologist, and it's my true pleasure to introduce another hematologist, Dr. Friederike Meyer-Wentrup, who's working at the PMC. And we had sort of quite similar journeys. We are both born in Germany, and we both made our first part of the education in Germany. And then you decided to be intrigued by the Netherlands, and you have been one of the founding members, I may say so, of the PMC, and really from the scratch started building this exciting new endeavor, and are now an integral part of their clinical care, but also, of course, of their science. And I think also an excellent person who's building bridges between those two centers. And you focus on lymphoma, and I'm more than excited to listen to your ideas over mature B cells and what this will be in the future in terms of cure of patients. Friederike.
Okay. Yeah, thank you very much, Jürgen, for this very kind introduction. The title of my presentation is Bridging the Gap, Translational Research for Advancing Pediatric B-cell Lymphoma Therapy. I'm a pediatric lymphoma specialist at the Dutch National Pediatric Cancer Center, the Princess Maxima Center for Pediatric Oncology. And we opened in 2018, and we are the largest pediatric cancer center in Europe. And we are not just a hospital, we are also a big pediatric cancer research institute. And all Dutch children with cancer are treated at our center. And per year we see about 250 new hemato-oncology patients. Pediatric lymphomas are actually a common pediatric malignancy. They are the third most common entity, and at the Maxima we see about 80 to 90 new patients per year. And most of these lymphomas are mature, B-cell derived, and for example, in 2023 we saw about 40 Hodgkin, 10 Burkitt and 12 other lymphomas. The diagnosis is made by immunohistochemistry and flow cytometry, and at our center it is always supported by DNA and RNA sequencing and also other molecular diagnostics. We apply a multimodal treatment that consists of chemotherapy, radiation therapy, immunotherapy, and in some cases also surgery. And the survival overall for our patients is very, very good for the majority. So if I sum this up, we see multiple lymphoma entities that are diagnosed, treated and studied by a group of professionals. And this multidisciplinary approach is reflected by the Maxima Lymphoma team. Our team is multidisciplinary and diverse. It consists of clinical care and clinical research colleagues, our colleagues from preclinical research, our partners from the UMC Utrecht, and also our partners from the laboratory diagnostics. And together we do research to improve the lives of children with lymphoma. I have the honor of chairing this lymphoma team together with Ruben von Boxtel, who is a group leader at the Maxima Research Institute dedicated to understanding why children get cancer. And he's an expert for advanced molecular technologies. When we started this group and started chairing this group, we were also wondering how we would want to work to make this a success. And we decided that we want to create opportunities for all the members. We want to be a truly cross-functional team, and we want to do our best to overcome the dichotomy between clinical and preclinical research. And we want to work in an agile way, where we collaborate rapidly based on our expertise. And I must say that this team that we have built really is the basis and the environment where I can do the research that I want to do to improve lymphoma care. So why do we do research on mature B-cell lymphomas? Overall survival is above 95%, but we see significant treatment-associated short and long-term toxicity, and the development of less toxic therapies is really a truly urgent medical need. So what we do with our research is we focus on toxicity reduction, and we do so by developing better therapeutics and also by developing biomarkers. And we think, and I personally also think this very strongly, that understanding pediatric lymphoma biology is a prerequisite to be successful in this endeavor. Our approach to connect bench to bedside and to really bridge the gap is what we would call our innovation cycle, where we start, for example, with performing molecular diagnostics and analyses like single-cell DNA and RNA sequencing. We move on to establishing cell culture systems to do in vitro studies. We develop novel therapeutics and move towards the clinics when we think of biomarker development, supportive care, and clinical trials. And for this cycle to be truly innovative, it's not so important where you step into it. It's more important that you try to really make the full circle. And I will show you examples where I think that we are almost there and we are doing our best to make this full circle to be innovative. When we started working on lymphoma, the first thing we really needed to do was to integrate the workflow for diagnostics and research. Because at the Maxima, we usually do lymph node biopsies to come to a diagnosis. And the diagnosis is made by standard pathology, so immunohistochemistry, and, for example, but also by flow cytometry and bulk DNA and RNA sequencing. And the single-cell suspensions that we use for flow cytometry are frozen viably and then stored in our biobank. And once patients and parents have given informed consent, this material is used for research such as single-cell DNA and RNA sequencing and also the development of novel culture systems. And the integration between what we do for diagnostics and research has really been crucial and has really laid the basis for the research that I can do and that I would like to show to you. Of course, I have not done this by myself, so setting up this workflow was really mostly the work of Lisette Westerer, a medical immunologist, Marijn Vermeulen, our pathologist, and Lennart Kester, who's a specialist for molecular diagnostics. In the course of my presentation, I would like to zoom in onto the research that we do on Hodgkin's lymphoma, which is the most frequent mature B-cell lymphoma we diagnose in children. And what makes Hodgkin's lymphoma so intriguing is its microenvironment dependence, and we think that this could really be the Achilles heel. You will see this cartoon that I have depicted here coming back at various moments throughout the course of this presentation because it nicely depicts the malignant cell, which is called the HRS cell, the Hodgkin-Reed-Sternberg cell, surrounded by the microenvironment cells. And what makes this lymphoma so intriguing is that the malignant cells are very scarce. Only 0.1 to 10% of the lymphoma are malignant, and the rest is a complex microenvironment consisting of benign immune cells. Overall survival of our patient is excellent, but significant long-term toxicity is a real medical problem. So we were wondering, can the microenvironment dependence be exploited to develop less toxic and more targeted therapies? Maybe this was a naive question, but when you are a young group, you can really afford to also ask these blue-eyed, rather naive questions. We were facing significant challenges. So the tumor cells are scarce, as I mentioned, and they depend on the microenvironment, and there were no in vitro or in vivo models to study this. So, together with Ruben van Boxtel, we started to think about this, and we decided that we wanted to start with single-cell RNA sequencing of pediatric Hodgkin lymphoma, and two very talented PhD students, Jurian de Kanter and Alexander Stemers, have worked and work on this. And what we did is we flow sorted the cells, the single cells that we had made during the diagnostic process, and sorted these and then did single-cell RNA sequencing. And what we call this usually the seed, which is the malignant cell, and the soil, and that's the benign environment. So I would like to take you through the findings of the RNA sequencing of the seed. So the Hodgkin-Reed-Sternberg cells encircled in red, so we could really isolate quite a significant amount of Hodgkin-Reed-Sternberg cells. And when we did the analysis and we compared our sequencing data to two data sets that had just been published, we could identify something which we call the Hodgkin-Reed-Sternberg cell core genes. And we analyzed these core genes, we had analyzed them in our samples, and we had two samples where we had a very high amount of Hodgkin-Reed-Sternberg cells, and we could really demonstrate the aggregate score of core gene expression in the HRS cells of these two patients. And some of the more important and other important findings were that we identified NRXN3 and LRP8 as novel potential Hodgkin-Reed-Sternberg cell surface markers. So these could be putative therapeutic targets. We also identified that IL-13 and its receptor are expressed in Hodgkin-Reed-Sternberg cells and could actually lead to an improved survival of the malignant cells. And we found a variable intratumoral expression of the core genes forming a continuum, as you can see here. We next went on to characterize the soil. So it's the same approach, but now we are zooming in on the benign immune cells. And what is interesting is that we could really isolate and analyze different immune cell populations. And what makes this data set interesting is that it also contains quite a few myeloid cells. And that is something that's quite rare because they are difficult to isolate and then to put through this pipeline. What we did, and again, Jurian de Kanter and Alexander Stemers did great work under supervision of Ruben. We looked at the presence and strength of HRS cell interactions with cells in the tumor microenvironment. So this is what is depicted here. And we compared this to the interactions we found in reactive lymph nodes for comparison. And doing this, we identified putative receptor ligand pairs that were where the interaction was upregulated in Hodgkin's lymphoma, and these could be targetable. To add spatial information to this, we performed RNA scope of selected receptor ligand pairs, and here you can see the co-localization of LAG3 on T cells with HLA class 2 on Hodgkin-Reed-Sternberg cells. Our most important findings here were that we identified CD69 and galectin-1 and LAG3 and HLA class 2 interactions between HRS and T cells in most tumors. We found that NK cells and germinal center B cells are depleted, and that most of the cells that are detected are exhausted T cells. So they are enriched and make up most of the tumor microenvironment. And I think the most interesting finding is actually that we found significant inter- and intra-patient heterogeneity of the HRS immune cell interactions. So there is quite a significant difference between individual patients, and this finding is something we want to further analyze in consecutive studies. While we were doing single-cell RNA sequencing, we also set up a workflow for single-cell DNA sequencing of pediatric Hodgkin-Reed-Sternberg cells. So again, we use the same pipeline. We have the single-cell suspension. It goes through FACS sorting. But to do these analyses, it is very important to have single Hodgkin-Reed-Sternberg cells. So not a single T cell sticking to these cells, which is difficult. And this cartoon or this picture, kind of shows the work, I would say, of probably 2 to 3 years optimizing and trying again. And this was also done by Jurian de Kanter and by Alexander Stemers under the supervision also of Ruben and myself. And what we ended up doing is putting these cells into agarosidomes, and the PhD students are now handpicking these cells and then putting the malignant cells into the whole genome analysis of single-cell pipeline so that we can analyze the data. And we compare with non-malignant B cells from the same lymphoma and use T cells from the lymphoma as germline control. So what this allows us to do is for the first time to understand the clonal evolution of pediatric Hodgkin's lymphoma. So we can follow the seed. We can look at driver mutations. So in blue, you can see the Hodgkin-Reed-Sternberg cells, and in red, the normal B cells. We also look at overall mutational load, mutational signature, and most intriguingly, we will be able to time events. So we will be able to say when, at which age of the patient a certain mutation occurred, and then compare that to the age of the patient when the lymphoma occurred. And this work is done again by Alexander Stemers and by Diana Petku, another very talented PhD student in our laboratory. When you start doing RNA sequencing experiments and you look at single-cell DNA sequencing, you will actually very soon realize that you need some sort of in vitro system to perform functional studies. And so we were wondering if we could generate an ex vivo Hodgkin lymphoma cell culture system. The challenges, again, big. There were no relevant in vitro systems to study the Hodgkin tumor cell microenvironment interactions. Primary Hodgkin lymphoma cells cannot be cultured because they depend on the microenvironment. And then the Hodgkin cell lines that are available, they grow without a microenvironment. So you may wonder what their value is in these studies. So at this moment, luckily, I got to talk to Victor Peperzak, who's a principal investigator and group leader at the UMC Utrecht. He's an expert for B-cell malignancies, and he's very much interested in difficult to culture B-cell malignancies. So we were using single-cell suspensions and started experimenting. And we found that the 3D GrowDex hydrogel culture system that he had established resulted in viable malignant and non-malignant cells after at least 7 days and up to 10 and sometimes even 14 days in culture. And what this allowed us for the first time was to do pilot compound screens with primary Hodgkin lymphoma cells. And what you can do is you can, for example, look at different BCL inhibitors. You can look what prednisolone does to your cells, and you can differentiate. For example, if you look at apoptosis, you can see that prednisolone does not induce apoptosis in B cells, but in Hodgkin-Reed-Sternberg cells and in some T cells. And we also find, for example, that Hodgkin lymphoma cells are very sensitive to BCL-X inhibition, and at that time when we had found that more to characterize our system, this was also published. So for us this was nice because we knew our system worked. Now, together with Victor, we developed a program and got support from KWF to study the interaction between the malignant cells and the CD4 T cells. So going back to the cartoon, this is an important interaction for the survival of the malignant cell. And what we want to do, and Diana Petko is the PhD student who has now been working on this for 1.5 years, is characterize the interaction, look what disrupting the interaction does to the malignant cells, and also see if we can inhibit signaling pathways to inhibit the interaction and to harm the malignant cells. When we move through the innovation cycle, we have started with the single-cell genetic analysis that I showed you. I have showed you experiments that we do with an in vitro or ex vivo culture system that we established. And I would like to move more towards the clinical application. So we were wondering if immunotherapy can reduce treatment-related toxicity in recombinant and in recurrent and refractory Hodgkin's lymphoma, because disease occurs in about 10% of patients and there is no standard treatment. And the treatment that we give is usually high-dose chemotherapy, which has a very good overall survival, but again, very relevant toxicity for our young patients. So we were looking again at the cartoon, and when you look closely, you see that there are targetable antigens expressed in the tumor microenvironment and there are therapeutic antibodies available. So when we got the chance to participate in the CheckMate 744 study, we were very enthusiastic to participate. We evaluated the induction of remission by nivolumab and brentuximab vedotin, and my colleague Auke Beishausen was our national principal investigator and led us through this study very successfully. And the results were published and they were very good. The low-risk arm and the high-risk arm had excellent progression-free survivals, and now also longer-term data has been published and it still looks very good. When the study was finished, we as a group thought, yeah, we cannot just go back to just offering our high-dose chemotherapy. That felt very wrong. So we decided to offer the immunotherapy to all our refractory recurrent Hodgkin lymphoma patients. And in total we treated 10 patients, and we found that complete remission was reached in all of them prior to consolidation, and all our patients are still in complete remission, and toxicity, most importantly, was very low. And we could add this as real-world data to the results of the CheckMate study. Now, where do we move from here? What are the future perspectives, and how can we de-escalate chemoimmunotherapy further? I've talked to you about targeted therapy, but sometimes targeted therapy can lead to the development of resistance, and added value is often in combination with chemotherapy. So we really think that treatment stratification based on biomarkers and detoxifying immunochemotherapy are interesting approaches to reduce treatment toxicity. The challenges are big. So we have a lack of reliable biomarkers and a very limited knowledge on optimal chemotherapy dosing and administration, and the ethics of de-escalation in view of excellent survival with current therapy are very important. So to address this, Ruben von Boxel and I developed the De-Escalate research program. What we want to do is look at cohorts in time and space to identify biomarkers for stratification. So we will look at patient cohorts from the Netherlands who were treated with less intensive chemotherapy and compare responders to non-responders. And we will do the same with cohorts from low-resource settings, where children are treated less intensively. And again, we will look at responders and compare them to non-responders. And we also want to look at data and samples from adult patients and learn from the adult experience. In a second step, we want to do in vitro and in vivo experiments to detoxify chemoimmunotherapy, look at different concentrations, like what I showed you in the Hodgkin model, where you can just titrate chemotherapies and look what the best amounts of chemotherapy, the best concentrations are. And we want to also test novel chemotherapeutic compounds. And finally, we want to do an in-depth analysis of the ethics of studies and shared decision making. And we want to also look at the risk-benefit sharing if we collaborate with parties from low resource settings. And with this, I would like to thank you for your attention.
Yeah, impressive and also inspiring. Thank you so much. Because I think you clearly showed, on the one hand, how you can, from really the scratch where you came, build up a complete network, patient samples, which was really not very easy. On the other hand, you showed clearly your love for the detail, I think, which is also very important to isolate one cell. But you find also back your partners at the campus, for example, with the 3D models, which really should be hopefully something inspiring for everybody listening here, how rich the campus is in potential collaborations. And if you have one unique ingredient, somebody else at the campus might have another unique ingredient. And I hope that with your new role you have now also in the spearpin cancer, you will mentor also the next generation to really search not only abroad, but also in particular here at the campus Utrecht for excellent partners. So thank you very much and welcome again.
Thanks a lot, Jürgen.